Knowing that decomposition can vary spatially and temporally, here we'll cover the factors that control rates of decomposition. Decomposition in ecosystems is controlled by three major factors. The first is substrate quality, the second, characteristics of the microbial community, and third, the physical environment. First, we'll deal with substrate quality, or here, litter quality. Now, on some scales, the availability of below-ground resources is the factor governing ecological patterns in litter quality. So if you want to understand whether a system is going to have high-quality litter, meaning that it's easy to decompose, or low-quality litter, you need to take a look at the below-ground resources, which in turn affect the characteristics of the plants, which then determines the quality of the litter. So plants that dominate high fertility sites tend to produce labile litter, high-quality litter. And if you look across different types of plants in different climates, you'll see what this pattern is. So for example, in a temperate climate, here are, on the x-axis, different classes of plants. Vines, herbs, shrubs, trees, grasses, different types of plants with deciduous leaves, and then those that have evergreen leaves. And in general, this is a pattern associated with fertility. So high fertility plants on the left, low fertility plants on the right. On the y-axis, we have dry mass loss, so percent of original in a given amount of time. So what's the rate of decomposition? How much mass is lost from looking at the decomposition of leaves of all of these organisms? So here we have leaves along a fertility gradient in a temperate climate, and here a semi-arid climate. Looking at the temperate climate, as we go along this fertility gradient, for species that dominate high fertility sites to low fertility sites, the dry mass loss decreases for a given amount of time. So dry mass loss means the amount of dry matter that's lost in a given amount of time. So vines which dominate high fertility areas can lose 75% of their mass in a given unit of time, whereas it's somewhere on the order of a third of that for these evergreen trees and shrubs. The same pattern exists in semi-arid climates. Not quite as strong, but still those species associated with high fertility sites down to low fertility sites decompose faster to slower. Now we can also see that when we take a look at three different classes of compounds or of organic matter. So first we'll compare the mass loss of a pine branch, so the branch of a pine tree, pinus, and then the needle of a pine tree, which is going to be higher, have a lot more nutrients in it and a lot more labile carbon than the branch does. And then lastly, a pin cherry leaf. So pin cherry is a relatively weedy tree, tends to have really high nitrogen concentrations in disturbed areas, for example. We're going to take a look at a leaf of that and compare it to needles and branches of pine trees. On the x-axis here is time, and this is in months. So here's 1 year and 2 years, and on the y-axis we have the mass remaining at that point in time. So initially it's 100%, and then we have 50 and 0. And you can see the pin cherry leaf, which is considered to be a relatively nutritious, high quality part of the plant, and pin cherry itself is a high fertility species, loses a lot more matter at any one point in time than the pine needle does or the pine branch. So this is a time course of decomposition of these three different types of organic matter. So it's important to know that there's differences in quality as far as decomposers are concerned for different classes of biomass. And as we covered before, the costs of producing that biomass are the same. So whether you want to construct a leaf or a root or a branch, for all intents and purposes, on a gram per gram basis, it costs about the same amount of energy to produce those. But there's big differences in how fast they decompose. Now, the carbon quality of substrates is the primary chemical determinant of the decomposition of fresh litter. So we really need to think about the carbon quality of those substrates if we're trying to understand how fast fresh litter decomposes. And when we look across substrates at different compounds, there's a hierarchy of lability. So there's some compounds that are more labile than others. So at first we generally think of labile metabolic compounds. These might be proteins, for example, that are associated with the actual metabolism, the cell contents of plants, for example. And then there's moderately labile structural compounds, and we talked a little bit about this, the celluloses and hemicelluloses that make up the structure of plant material, of cell walls, but are relatively labile. So good examples of these are cellulose and hemicellulose. Now, lastly, we have recalcitrant structural material. So these are also involved in cell walls, for example, but they're very hard to decompose. And lignin is a good example of this, but also waxes that make up the cuticle of leaves. Now, when we talk about chemical lability, we talk about a couple of things. First, there's the energy content of bonds between different atoms. And when you have high energy bonds, so for example, a carbon-carbon double bond, these are relatively difficult to break compared to a single bond. And the breaking of these bonds is associated with the oxidation, the respiration, the energy gain for decomposing these compounds. So if you have high energy bonds, if you have compounds that have high energy bonds, those are the ones that are going to be most difficult to break and the most difficult to decompose. In addition, a compound can be difficult to decompose if it has a relatively irregular structure. So enzymes are usually used to break down organic matter and turn recalcitrant organic matter into smaller, more labile organic matter that can be taken up by the cells and then used for respiratory purposes. But some compounds have a very irregular structure, and as a consequence of that, enzymes don't work that well. And so instead, they have to be broken down non-enzymatically. So for example, there's classes of bacteria that produce peroxides that non-enzymatically can just blow apart the organic matter that's in soil and then can use the byproducts of that reaction for metabolic purposes.